Well, I have the distinct pleasure of introducing our speaker. Not sure why I'm doing this job, I guess, because Zach told me so. But I was the person I invited Steve to give a talk because, as you are very aware, he is the VP RGS of MSU, right? And he happened to do accounting science work, which is very intriguing to me, you know. So I was just curious what he does. So I can tell a little bit what he does. I read his CV, very impressive.
This is to satisfy your curiosity.
Right. Okay, Steve got his bachelor's degree in physics from Caltech, and then went on to Berkeley, earned a master's and a PhD degree in physics. Then he spent some time at Yale as assistant professor, then moved to Oregon, which seems that he went directly from assistant professor to full professor. Wasn't that case, but he has actually, in addition to his VPRGS title at MSU, which actually I found a little regret because that's how we announced this talk, that Steve Shu is the VPRGS, but he actually is a professor of physics too at MSU. Speaking of that, I'm not sure if you know what VPRGS does, which actually I studied a little bit. Well, actually, it's in his CV, which is easy. But to summarize, it does everything you care about, like tenure, promotion, all right, graduate school, regulatory affairs that include RD and animal use, commercialization, MSU intellectual property that has startup, you know, business stuff, MSU Foundation, of course. So I guess everybody will behave well today, right? Don't ask too many more questions. Is that right?
No, I want people to ask whatever questions they want.
Yeah, sure. I couldn't help that. Okay, so his main interest is in theoretical physics, quantum field theory. So I think that would be perhaps one of the first. Would somebody who knows quantum field theory give a talk in this room? Hopefully he won't talk about quantum field theory because I'm pretty sure nobody will have a clue.
Physics building.
I'm getting there. So, yeah, you actually, but he has, sorry, this is getting a little too long. But he has over 100 publications mostly in theoretical physics. Speaking of physics, as Francis pointed out, I don't even know this building used to be physics. And that street is called Physics Road. So, you're actually in the right place right today because we are right in the intersection of physics. That street sign is right over there. For me, this is very satisfying because I do actually psychophysics, which is a branch of psychology, but not too much physics. So this is very welcome home. I actually have some idea of adding another thing over here, making it a little more cooler. I see some friends are laughing. I'm crazy. But if you're interested, we can talk about that later. But that third street, there's another street over there.
Well, what are you going to add?
Oh, no, it's not. So, okay, so he has. Oh, okay, so he's a professor of physics. He's also an entrepreneur, interested in internet security technology. So he had co-founded a company called SafeWeb, was sold to Symantec, which is runs the antivirus software on the computers, I guess for a lot of money. He also co-founded another company called Robot Genius, so he's also an entrepreneur. He has done all the things, testified before Congress, given many lectures at different academic institutions and also businesses. So that's all, maybe not most relevant to today's talk. I think today he's going to talk about his third interest, which is cognitive genomics and bioinformatics. He actually helped founded the BGI Cognitive Genomics Lab, which we probably talk about. It's a big, currently is a state key lab designated by the Chinese government, which is, I think that means it's a big operation, a lot of resources, a lot of money. Yeah. Thank you. Please welcome Cecil.
I won't be charged for that time, right? I think I may have given a talk in this building because I gave a colloquium in the physics department here in the early 90s. So I don't know about this particular room, but I think I've been in this building. Now, I enjoy giving talks much more when people ask me lots of questions. So I welcome questions at any time. Just put your hand up or yell at me. And I'm sure I won't be charged for the time that it takes to answer the question. So, now I have a lot of slides here. I'm covering a lot of material. Some of it will be probably very familiar to people in this audience, and some will be less familiar. The slides are available online, so at some points when I'm just trying to move fast, I will just sort of indicate what's on the slide and go past it. And if you want, you can always stop me and I'll spend more time on the slide, or you can go away later and look at it online. Typically those will be slides with lots of detail or data on it. Okay, so title of my talk is Genetic Architecture of Intelligence and Other Quantitative Traits. If there's maybe one thing that I want you to take away from this talk is that, in my opinion, we're right at the threshold of being able to actually learn specific things. Which button turns on? Oh. Learn specific things about the genetic architecture of something as complicated as intelligence. And that's actually a bold claim, but we'll see at the end whether you believe me or not. Okay. As mentioned, my main field of research is actually theoretical physics. We'll talk about theoretical physics today. When I accepted this VP position, I had no idea how painful administrative stuff was going to turn out to be. But I did manage to write one paper in the time that I've been here at MSU. So I'm actually, my colleagues in physics were shocked that I could actually write a paper while I was doing this job. I surprised myself, actually. Okay, so why is a physicist interested in genomics and intelligence and bioinformatics? This slide, I think, does a reasonable job of explaining the interest. This is a log plot. It shows an exponential decrease in the cost of sequencing. This is, for comparison, Moore's law. All the revolution in computer science, in internet functionality, our ability to store data, that's all driven by this curve. And you can see that the decrease in cost of genotyping is decreasing even faster, really much faster at a super exponential rate. And if this curve continues, then we'll literally be drowning in genomic data. And the question is, what can we do to make use of that? And I believe it will actually impact directly on fields that people in this audience are interested in. I should also say that the questions about human intelligence and questions about genetics and evolution, those are things that I've been interested in really since I was a kid. And so I think I can honestly say I've been interested in those things longer than I've been interested in physics, because you can ask questions about those things at quite a young age without actually even knowing anything about physics. However, I didn't really think as a kid, other than in science fiction stories, that we would be able to do anything really substantive in this area. And it wasn't really until I started seeing this curve that I thought, well, maybe in my lifetime we're going to be able to actually do some of this work. And now I actually think we're going to be able to do some of this work in the next 10 years. Okay, so a little primer on human genetics. It may not be necessary for this audience, but if nothing else, it will illustrate to you the crude way in which physicists think about other people's fields. I think of a genome as a list of about 3 billion base pairs. Humans are very similar, so at most of those base pairs, we're identical. If you choose 2 people out of this audience, it's only about 1 in 1,000 places in the genome that we differ from each other. Nevertheless, all the human variation that we see is caused by, or at least partially caused, by that small amount of variation. Thinking information theoretically, because the variation rate is so low, it means that this information is actually highly compressible. You can compress it actually down to a few megabits if you want per person. So if you needed to store the genomic information for all the humans on the Earth, you can easily do it right now. Okay. You probably are familiar with this term, SNP, single nucleotide polymorphisms. rather than look at all 3 billion loci in the genome, you could instead choose some particular subset of sites, let's say about 1 million, where there's actually common variation, where it's likely for two people to differ. So certain places are sites where there's tends to be a lot of variation between individuals. And so you can think of the SNPs as a subset of these loci and a particularly informative sampling of the whole thing. And this is cheaper to read out. So to read out the SNPs for a person, it's about $100. To read out the whole genome, it's about 1,000. Now these numbers are not exact, so again, physicists like to usually think in terms of orders of magnitude. This cost is really more like $200 or $300, and this is really potentially a lot more than $1,000, depending on what level of coverage you want. Sequencing different error rate. Now, throughout this talk, mostly I'll be talking about SNP data, although we actually are getting full whole genome data on some of our stuff right now. But I want to emphasize that a few years from now, these cost curves are going to cross. This is not really getting much cheaper, but this is getting cheaper at a very fast rate. So graduate students in the future will often come to your office and ask, what were these SNP things they talked about back in the old days? What exactly is that? So, because they'll have access to the whole genome. Okay, so here's a outline of what I'm going to talk about. Part one is probably very familiar to you, although it isn't without controversy, this subject of whether we can measure people's cognitive ability. I'm going to try to go through that relatively quickly. The main thing I just want to establish is this is at least a reasonable phenotype to look at. If you really don't like human intelligence as a phenotype, there are many people who really hate the idea that that could be a human phenotype. Just replace, every time I use the word cognitive ability or intelligence, just replace it with the word height. Because as I'll show you, in terms of the parameters, the things that I care about in this talk, they're basically the same. There are small quantitative differences, but there do not appear to be qualitative differences between intelligence and height. I'll talk about a GWAS that we're doing. This is a project with BGI, which is a big genomics lab in China. And in the end, at the end, I'll talk about something really specifically dealing with the genetic architecture of both height and intelligence. Okay, now this is obviously an interdisciplinary subject. You can see some very, very interesting topics of exploration here. You can see the names of the different fields, the names of the groups of people who study these topics. Everything I'm going to talk about today, or primarily what I'm going to talk about today, is really a cheap statistical hack, which basically just tries to read out the genetic information of an individual, look at the particular phenotype, and look for statistical correlations between the two. So conceptually, it's really very simple, what I'm going to talk about. However, the output of that kind of study is extremely interesting to all the people who work in these other circles. So for example, if one finds a particular gene that is associated with height or associated with intelligence, immediately people are going to ask, what's the protein that codes for? Is that some interesting pathway in the brain? People are going to ask whether there are drugs that can be built around that functionality. People who study evolution are going to be asking about whether that gene has been under selection, et cetera, et cetera. So you can imagine that although what I'm doing is really, in some sense, conceptually very simple, it's a particular direction where clearly we're going to make a lot of progress, and that progress will have impact broadly throughout science. Okay, so what minimal properties do you want for a quantitative phenotype? Well, you'd like it to be stable and be something you can measure reliably. Okay, so if I measure your height today, it's a little bit of variation in height, maybe 1 or 2 centimeters throughout the day as you get tired. But generally your height doesn't vary very much, and unless there's some really horrible accident that you're in, a year from now your height will be pretty much the same as it was when I measured it today. So I'm going to argue that the measures we use for cognitive ability are also quite stable. I'll show you some data about that. We'd like the thing that we're measuring to have some quote validity. Maybe it has some predictive power for life outcomes, or measure something obviously real like the physical size of an object. And finally, you'd like it to be heritable. So if it's going to have some connection to genes, hopefully it's something which is actually influenced by genes. So what I'm going to argue first in just this first part, maybe I don't need to argue this in front of this audience, but that for cognitive ability, all of these conditions are actually satisfied. Okay, so how does a physicist think about G for IQ or general cognitive ability, general mental ability, whatever you want to call it? Well, here's how a reasonable person might approach this question. Well, we all believe that there's some sense in which people are better or worse in terms of cognitive ability. But a priori, you might think, well, there are many different types of cognitive abilities. You might first start by inventing some battery of cognitive tests. There are obviously things that you would put in here, like the ability to recall things, mastery of language, ability to think quantitatively, ability to think spatially. These, I think, would be all uncontroversial components of cognitive ability. You can also talk about slightly more controversial ones. We can talk about reaction time or something like image recognition. Historically, these actually play an interesting role in the history of G. But probably you'd get a lot of argument from people about whether we should be really included in the list. But certainly we can come up with some relatively controversial list of tests. And then, just thinking empirically, we could go out and just test a lot of people. And so we'd get a map from each individual to some n vector. That's just a list of these scores. But the interesting question is, how do you get from that n vector per individual to some kind of single number measure of cognitive ability? When you read in the popular press about IQ scores, they just seem to give one number. So how do you get from this n vector, which probably nobody really complains that measuring this n vector from an individual is not a bad thing to do? How do you get from that to this single number? And amazingly, I consider this actually a really important discovery of modern science, but apparently most people are unaware of the actual result. The result is that there's a tremendous amount of intercorrelation between those n different abilities that I mentioned on the last slide. And in fact, people don't uniformly fill out the space of possibilities of that n vector. They actually fill out a subspace, which is much smaller than the full volume. And that subspace looks kind of ellipsoidal. And so if you want to compress this information, what you do is you use factor analysis to find the largest principal component of this ellipsoid. Here, this is actual data from Project Talent. So that's 100,000 ninth graders that were tested, and they were given mathematical, verbal, and spatial tests. And you can see they fill out this sort of, this is a volume, but they actually fill out just this ellipsoid inside their 3D cube. And if you wanted to compress this information, instead of giving the actual triplet of numbers for each kid, you could just project them onto this axis and give their location along this axis. And that's really what's done when people extract the G factor or the IQ score. This is actual SAT math, SAT verbal data. And so if you think about this data, you can describe it in the following way. If all I know about somebody is that they have a high math score, just that fact alone increases the likelihood that they also have a high verbal score. Okay? And similarly with verbal and math. And that actually goes against a lot of our folk wisdom. So if you talk to most moms at the school where my kids, or sorry, dads as well, they'll often say things like, oh, well, Johnny's good at math, so he's not going to be very good at verbal stuff, which is actually completely wrong. If Johnny, all I know about Johnny is that he's good at math, the chances are he's also pretty verbal. Now, of course, compressing all this information down just to a coordinate on this axis, you lose some information. So there are people who are good at math and not so relatively good at verbal. Then there are people who are good at math and really good at verbal. So there's quite a lot of, this is a lossy kind of compression. To take that whole n vector and just replace it by a single number, you're losing some information. But it's not a crazy thing to do if all you want to do is compress it down to a single number. Then the real question is whether that single number has a predictive ability or whether it's stable. Okay, so I think it's a reasonable thing to do. You all know that by convention, this G factor, which is often referred to as IQ, it's conventionally, it's normally distributed in the population. The mean is taken to be 100, standard deviation 15. Very similar to SAT scores. SAT, the mean is about 1,000, and the standard deviation is about 2. Okay, now you can complain about any of those n tests that I made up. You can complain that the vocabulary word I used was regatta. And of course, all the rich kids know what regattas are, and the poor kids know what regatta is. And so one goal might be to come up with a replacement for that battery of n tests, which is culturally neutral. So here's one called progressive matrices. You can think of these as time series or time snapshots at different times of an object which is evolving. And you try to intuit some rule for how these objects are meant to evolve, and then you try to guess what's in this left square. There's some multiple choice answers. And really, this seems pretty culturally neutral to me. It's basically testing your pattern recognition or your algorithmic reasoning ability. The surprising thing maybe is that the scores on this test correlate about 0.9 with the G score, with your location on that major axis. Okay, so one could, as a shortcut, if one didn't want to administer all of these tests, one could estimate the G-score of somebody by just giving them this much simpler test. You can give this test to people who can't even read, and so maybe that's a good way of estimating the G-score. Now, when something like this very primitive progressive matrix score correlates so highly with this mathematically very complex G-score, which is coordinated along a major axis or principal component, when you find that, it makes you start to think, well, maybe this G-score is some kind of window into the actual brain. It's actually measuring something kind of really deep about the brain. And here's some data getting back to this question of reaction time, which you might find interesting. So these are two populations of kids. The purple people are kids who scored really high in the SAT. This was all done in Boston, so these are actually probably Harvard and MIT students. These are students who are also pretty good scorers on the SAT. They're college students in Boston, but they're not as strong. And so this is just kind of illustrating the correlation between, this is Raven's progressive matrices, this is a vocabulary score, and this is how the two different groups segregate in this space. Now, what this psychologist is one of my collaborators, what he did was, he was interested in number sense. There's a French guy called Dehaene. Is anybody here familiar? Okay. So a former mathematician who's interested in number sense. And what the task here that was asked of the students is, they're shown a number on the screen, and they're asked to hit, I guess, one button if the number is below 45, and to hit another button if the number is above 45. When the number is far from 45, it's quite easy. Your brain processes it in a very quick way. It somehow can just sense that that number is very much less than 45 or very much greater than 45. So the reaction times are all lower. As the number gets closer to 45, your brain actually has to do some work, so it's a little bit harder to think a little bit about whether it's greater than or less. So you can see this, so that's why you get this general kind of mountain shape. But what's interesting is that in a very statistically significant way, you can separate the, quote, super smart or super high scoring kids from regular kids, and there's actually systematic reaction time differences. So, and this is a very old result, this reaction time correlation with G-score. If you are some of the people who work in this area that I know I refer to as G-men. And those are people who take what I call the strong interpretation of G, which is that it is really getting at something very fundamental about the goodness of function of your brain. And they would cite evidence like this for that. I don't necessarily find all that evidence convincing, and I don't really I don't consider myself a G-man, but it's an interesting hypothesis. Okay. About this time, someone will usually say something like, but can't you just study for the SAT and raise your score? How can it be telling you anything fundamental about your brain? So here's a very large statistics study with thousands and thousands of kids. This horizontal axis is the PSAT score times 10. The PSAT score is basically a shortened version of the SAT that you take like your junior year. And you can see the PSAT predicts the SAT score quite well. The students have been indicated in two different ways. The ones that are black received systematic coaching, so coaching that costs thousands of dollars, tutors, Kaplan's, Princeton Review, whatever you want. There is basically no difference between the kids who received the coaching and the ones between taking the PSAT and the SAT and the coached and the uncoached students. If you want to break it down in numbers, it's a fraction of the population, the standard deviation. So you can raise your score slightly, but on average, people do not raise their scores by very much by prepping for these tests, despite what these commercial companies want you to do. Okay? So you can think of this as evidence for the stability of the score, because it's just not very easy to change the score. Now, for a given kid, of course, there could be a kid who scored better on the PSAT than on the SAT, but population-wise, the PSAT is quite a good predictor of the SAT, and coaching doesn't seem to affect that. Okay. So, okay, what good is it? Well, for me, when I look at data like this, to me it's the most impressive quantitative stuff that I see in psychology. That I may be biased in saying that. Test correlation 1 year apart is typically 0.9 or higher. I'll talk about validity and I'll talk about heritability in the following slides. I did a bunch of work with a colleague just for fun on to what extent one could use SAT scores and high school GPA to predict college performance, and I won't talk about that now, but if you're interested, the quick summary is that SAT is correlated about 0.3 to 0.4 with measures of upper division performance in most majors. Okay. Now, as a physicist, I'm kind of interested in the far tail of intelligence. So what is that? Plus 2 standard deviations, that's an IQ of 130. That's sort of the top few percent of the population. Plus 3 standard deviations, that's 1.5. That's about 1 in 1,000 people. Plus 4 standard deviations, that's about 1 in 30,000. And if you've ever read Malcolm Gladwell, you know that, well, any IQ points you have above 120 are useless. They don't actually help you. That's established very rigorously by Malcolm Gladwell. And he's the researchers that he cites. Or the alternative hypothesis is, well, no, there are continuing positive returns to intelligence above some arbitrary threshold like this. And the answer to that question is quite interesting to me, especially as a physicist. So there's some data on this. There's a woman named Natalie Rowe. I don't know if anybody here has ever heard of Natalie Rowe. She was a psychologist at Harvard a long time ago. She wrote a book in which she randomly selected some eminent scientists. These are people in the National Academy. I think 1 or 2 of them later won the Nobel Prize. She selected them randomly across many different fields. And she was not a psychometrician, so she just basically went to interview them, asked them about their childhoods, and had them draw pictures of horses and stuff. But she had a colleague who was a psychometrician. And so one chapter of the book is devoted to some pretty high-ceiling tests of these individuals. And the mean score for this population of 64 eminent scientists was really quite high, especially when you compare it to the average for science PhDs. So why is it that the eminent ones do so much better than the average ones? Now, you can make up all kinds of stories about, well, it's not really causal, and the fact that they were smart had nothing to do with them being eminent. And it's maybe true. But I mean, maybe that this isn't completely conclusive, but it is quite suggestive to me. Here's some much better data. So this is from the study of mathematically precocious youth. There's also a sister study called the study of verbally precocious youth. And gifted education in the United States has gotten, in a sense, kind of standardized. So if your child is identified as gifted in school, like in the top few percent on some standardized test, the kid will often be encouraged to take the SAT early. And so that's a sort of standard high ceiling way to test these kids. And so there's a lot of data now. In fact, I would guess maybe a significant fraction, like a quarter of gifted children these days in America now take the SAT at a young age. And when you take the SAT before the age of 13, the ceiling is extremely high. So because they dumbed down the SAT in 1995, the best you can do as a senior if you get a perfect score is you're about 1 in 1,000 population. But if you take it as a 13-year-old, you could, if you get a perfect score, you're probably more like 1 in 100,000 or 1 in a billion. So the ceiling is much higher. So they've assembled, these researchers, Lubinsky and Benbow and others, Julian Stanley, I think is another one, they've assembled quite large cohorts of very intellectually gifted kids. And since they start with a population of kids that are sort of in the top few percent of the population, but they have the top group, which are really high up there, like 1 in 10,000, they can actually look at differential performance over their lifetimes of these kids. All these kids are gifted. All these kids are in gifted programs. All of them are from affluent families, effectively. And yet there are differential outcomes based on IQ above the top few percent threshold. In fact, if you compare the really top group, the 1 in 10,000 group, to NSF fellows at top 5 departments in electrical engineering or computer science or physics, these kids who are filtered just on the basis of a simple test at the age of 13 outperform the PhDs from MIT in terms of career success. So probability of tenure at a top research university, patents, et cetera, et cetera. So I call that validity. Give a kid a test for a few hours, it predicts life outcomes better than in some really sophisticated selection process, which happens much later in life, et cetera, et cetera. Okay, let me talk about heritability. Now, I have to say this so I don't get in trouble. Heritability is defined relative to a specific range of environments. Okay, so it's well established that it's obvious that if I make the range of environments large enough, the heritability could go to 0. Okay, so regardless of what your genetic gifts are, if I refuse to feed you, the correlation between your height and your genetic makeup is going to be reduced almost to 0. Even if you have genes for high intelligence, if I hit you in the head every night and keep you in a dark room, probably you're not. I'll be able to decouple your adult IQ from your genes. Almost all the data with one well-known example, maybe you know this example, by, which is this famous paper by Eric Turkheimer. Almost every example where heritability is measured in what I call using classical methods, there's a restricted range of environments. So there are typically families that are at least good enough to be allowed to adopt kids. Okay? And that's pretty much a, in one sense you can say that's a pretty narrow range of families. But on the other hand, you can say, well, that includes coal miners who didn't finish high school and people with couples with 2 PhDs. So that there's still some range in there. Okay, so it's not zero range, but of course we're not including real serious deprivation. Okay? So that's an important hypothesis. So here's some data. IQ correlation for monozygotic twins reared apart, IQ correlation for monozygotic twins reared together. You can see that the correlation is high, so monozygotic twins are quite similar. And they're quite similar whether they were reared in the same family or they were reared in, the same family or different family. You could think of this reduction in correlation as the effect of the environment. It's not very big. And you can see the same thing here for the full six. So this is all, people fought apparently like cats and dogs over this in the 70s, but now I don't hear a lot of people complaining about these results. Just to show you that there are really, really large sample size results. This kind of thing's been replicated quite a bit. One of our collaborators, Robert Plomin, has access, I think, to like every team in the UK to the national health system. So, this is just not it's not a really controversial result that the heritability of intelligence is quite high. Okay. Now, I refer to those as classical methods because they're now different and new methods for measuring heritability. So, rather than look at special cases, so special cases meaning I know that these guys share all their genes, or I know that these guys share half their genes. Those are sort of discrete point cases that nature gives us. Rather than doing that, I can just pick random people from the population who are seemingly unrelated, although secretly, if you go far enough back, we're all related. But they're seemingly, not knowingly related people. I can measure the IQ difference between a pair of such people, and I can measure the genetic distance or difference between those two people, and I can regress one against the other. It's effectively, if you think about it, what we're doing over here. But we have to do it here with special cases that nature gives us access to. Nowadays, I don't have to do that. I can just grab a few thousand unrelated people, and I can try to measure for any trait, any phenotype that I can measure on that population, I can try to deduce the heritability. And it's an interesting question whether the result deduced this way is consistent with the classical result, and the answer is that it is. Completely consistent. Now, this is a cartoon. This is not real data, but this is just to emphasize what I'm saying. So here's the level of genetic relatedness. Maximum value is 1 for monozygotic twins. Here's the correlation between IQ scores. And typically for adult twins, you might get a value as high as 8 or something. Full SIV, you get something just kind of half of that. This might be a cousin, or I don't know what it was here. But what are these little blue dots? What are these little blue dots? These are people who are not knowingly related, but they are either more or less genetically similar to each other, pairs of people. And we can measure whether, to what extent, having a pair that are more similar genetically makes them also more similar on the phenotype, in this case intelligence. And if you fit this data, you get results from this method which are consistent with that method. Okay, so in the early days there were all these people complaining about, well, it's all because the in utero environment was the same. That's why the twins are alike, because they share the same uterus. Well, these people did not share the same uterus. So, okay, but this is a modern method. Now, if you're mathematically inclined and you actually know some population genetics, and you see a linear thing like this, it immediately makes you think about what actually is going on with the underlying genes that would allow such a simple relationship. Like, as I vary the fraction of genes you have in common with another person, the similarity sort of just increases linearly. Not non-linearly, not like a cube, not like a quartic, but linearly. So what underlying model of genetics would actually be consistent with that? Well, it turns out that this kind of genetics was once derisively referred to as beanbag genetics, but it actually seems to fit all the data. So you can actually model all this data by just saying that of all the 3 billion loci in the genome, there's some subset which affect the phenotype. And each particular locus, let me call those the associated alleles or the causal alleles. For each of those causal alleles, I associate an effect size, which can either be positive or negative. Okay? And basically, to characterize someone's, quote, the genetic component of their IQ or their height, I just have to list all of the values. So plus 0.1 standard deviation, plus 0.01 standard deviation. So these are all different loci, and they either have one variant or the other at that locus. And I just sum that up in a linear way in order to get the prediction for the phenotype. And of course, if you randomly sample from distributions like this, you'll get a normal distribution. So it fits everything that we know about that. It's remarkably simple. Okay. Now, why are phenotype differences linear functions of genotype if, in fact, when you go in the lab, you can actually find gene-gene interactions, you can find all kinds of nonlinearities. For example, you can find examples where the effect of little c is not twice the effect of having just one copy of the variant. So there are all kinds of complex interactions that are known. That often is referred to as epistasis. But why would it be that when you collectively try to estimate the total phenotype of an individual, you get this simple linear relationship? Well, that can be explained if the variants, little c, are relatively rare. So suppose in 90% of the population is big C, maybe 9 or 10% of the population is little c, and then only 1% of the population is little c. If this variant little c is rare, then even if there's a nonlinear relationship between the effect of little c, and this other outcome, that will be suppressed in the effect of the data that we see. Okay? So a high degree of nonlinearity at the genetic level can still correspond to almost linear aggregate variation between individuals, if in fact the causal variance is really rare. So that's an open question whether causal variants are going to turn out to be rare, and I'll show you some data about that later. Okay. There's another reason to expect linearity. See, nonlinearity arises from things like this gene is controlled by this gene, which is far away, or this gene interacts with this gene, which is far away. Okay? But that's a very bad unit of selection. That's a very bad unit of design for nature to select on, because when I mate, my DNA gets scrambled up. I go through recombination, and my kid may get this chunk from me, but this chunk may come from my mate. Okay? So there's a basic theorem in population genetics, which goes all the way back to Fisher. And what it says is the rate of response to selection is actually controlled by the additive variance of the population. This is the additive part. Because I would much rather have a unit of selection or a unit of design where the effect of version A or version B here just is plus or minus independent of everything else that happens out here. Because all this stuff is going to get mixed. Okay? So in fact, in the limit where, so for sexually reproducing species, if the recombination time scale, that's 1 generation, so 20 years or something, is smaller than the evolutionary time scale, which could be thousands or millions of years, as long as you're in this limit, this is Fisher's theorem. Okay? And what it says is evolution has mainly been acting on beanbags. It's been mainly acting on these pluses and minuses. It cannot really do very much with things which are horribly nonlinear and coupled. Okay, so that's a theoretical reason why you might expect to see this linearity. So if I were to write this out mathematically, I'd say, well, general model for quantitative phenotype is, well, phenotype is given by some genetic information dotted in, combined with some effect sizes. But of course, if I have nonlinear terms, there could be a sort of tensor which links, with which the genomic information appears twice or 3 times. Okay. But terms like this will lead to deviations from that straight line that I showed you in the cartoon. And so, to some approximation, we think that we can get away with just that. And so, my prediction is that, if you look at this as a mathematical problem or a statistical problem, I predict that for many quantitative phenotypes like height and IQ, we'll extract most of these effect sizes within about the next 10 years. It's just a matter of accumulating that data. And once you do that, you'll have a predictor, so you'll be able to, if you read off the genetic, these X's are then known. If you read off the G for a given individual, then you have a prediction of the phenotype. Okay, let me talk about GWAS that I'm involved in. So everybody here knows what a GWAS is, right? Genome-wide association study. This is a nice review article from Plomin. It's already a little bit out of date, but basically there's been very little success so far in finding genes which are linked to intelligence. At the genome-wide level of significance, so far there are no hits. Okay. Contrast that with height. For height, there are currently, actually this is also a little bit old now, but there are about 750 genes found so far correlated to height at the genome-wide significance level. And these generally replicate. So if you find one of these genes, or loci, or SNPs actually is the right way to say it. If one of these SNPs is associated with height in, say, the European population, you do it on another population, you do it on a population of Asians, you find it's also associated. So these are pretty robust. And that captures maybe about 20% of the total genetic variance. So there's still some missing heritability, but we have a long way to go. And so what's interesting is, well, what's it going to take to find the first genome-wide significant IQ hit? Here's a graph which shows the history of GWAS for a number of different traits. Height is these red dots. Discovery sample size here is a log plot, so it's varying from 2,000 to 20,000 to 200,000. And now in the can they have, probably approaching 500,000 people with heights in SNP genotypes. But the history is interesting. So for a long time, nobody could find any genome-wide significant hits for height. Wow, missing heritability. How can it be? We know height is highly heritable from classical twin studies. How can it be we can't find a single hit? There must be something deeply wrong with our understanding of genetics, et cetera, et cetera, et cetera. Okay. But then, of course, once you hit a certain sample size, you start discovering, you start getting hits like crazy. Okay. And I predict it's rather obvious. We're going to follow the same pattern with IQ. But where are we with IQ right now? We're like here. Okay. So here's a paper from just a couple weeks ago. This study had about 18,000 individuals in it. Amazingly, the way the privacy rules work for IRB, you cannot pool this data and analyze it the way you should analyze it. You have to what you can do is compute some summary statistics from a chunk. The biggest chunk in here is maybe 5,000 people or 7,000 people. So you can compute summary statistics from a chunk, and then you can combine that with summary statistics from other chunks. But amazingly, no one's allowed to actually pool their data. Okay. Well, what do they find? Well, they find a number which is consistent. Heritability of childhood IQ is a bit lower because when people are young, typically the parents control their environment, but as they get older, they control their environment. So when you're young, even the classical studies tend to give a lower value for the heritability. And here you get a value which is consistent with those classical methods. There's one interesting candidate for a hit. It's not significant at the genome-wide level. For the genome-wide level, what you really want is p less than 0.05, but then there are a million SNPs you're looking at, right? So what you really want is 5 times 10 to the minus 8 significance for it to be genome-wide significant. This is not genome-wide significant. However, this one has been found as a prime candidate, both in this study and then an earlier one also with Old family. So this one, if you had to bet money, this maybe is going to turn out to be a hit once we get enough sample size. Okay, but it just shows you where we're headed. Okay. So what are we up to? I'm working with this big lab in China. It has significant resources due to this strange situation in China, which is sort of this crony state capitalism situation where some people can lay their hands on just gigantic chunks of money and resources. They have hundreds of Illumina ICs, so they have just this incredible platform for doing sequencing. In the old days, so this used to be called Beijing Genomics Institute. It used to be a government institute, but now it's private. When they were a government institute, they were a small player in the Human Genome Project. They did about 1% of the reads in the Human Genome Project. But since then, they've done a lot of interesting stuff. They did a lot of the early hominid sequences, like the Denisovans and Neanderthals. The most interesting thing I think they've done is Tibetan altitude adaptation. So if you look at Tibetans, they are very similar genetically to Han Chinese. But all Tibetans have a certain set of mutations which adapt them to life at high altitude. Only about 5% of certain Han Chinese in the surrounding area, lowland areas, have that mutation. And if you look in the genetics, it looks like basically over 5 or 10,000 years, these Han Chinese, this is in the distant past, started migrating up the mountain, and the ones that had the mutation out-reproduced the ones that didn't have it. For example, miscarriage rates for people who don't have a mutation are much, much higher at high altitudes. So this is an example. This, as well as lactose tolerance, are 2 examples of human evolution, very well established at the genetic level, and took place over the last 5,000 or 10,000 years. So real quantitative evidence of human evolution and happening on timescales which are almost within recorded memory. I mean, 5,000 or 10,000 years. So to me, that's intellectually some of the most interesting stuff. Okay, this is one of their buildings in Shenzhen. These are crates of aluminum machines. This guy's named James Lee. Some of you might know who he is. This is a list of top 10 institutions in Nature Publishing Index for China. And you can see, although BGI is a single lab, it's not a university. It's up here, just beat out Beijing University and all these other schools. So it's pretty impressive. Nevertheless, despite this kind of success, it's in a very gritty area. It's right across the border from Hong Kong. These are cranes. This is a big container port. If you walk out on the street, you can see chickens. And, it's not. It's pretty gritty. Okay. These are row upon row of cubicles full of bioinformaticians. These are row upon row of $500,000 aluminum machines. Okay, so the Archie-WAS is a little bit different from one where you just grab people out of the ordinary population. We're trying to do a case control design, so we have a high group and a normal group. And originally, the goal was to get thousands of people who are at least +3 in IQ, so about the 1 in 1,000 level. But we ended up hooking up with Robert Plomin, and he has this sample, which he got through SNPY and SPPY. These are people who are now adults, but the oldest ones are like 50. But they scored at this 1 in 10,000 level when they were children, so it's a very high-end sample. They have like typical IQ in the sample is probably more like 160. And I think I'm told there's at least one Nobel Prize winner in the population. Okay, so we have DNA samples from these guys, and we're right in the middle actually now doing not SNP sequencing, but whole genome sequencing on this one. Okay, why is it better to have a relatively smaller number of people, but people who are very exceptional? Well, you can do a statistical analysis. The point is, because these people are cases as opposed to controls and have a very high value of the phenotype, you get significantly more statistical power. So this group of 2,000 gives us roughly about as much statistical power as maybe 20,000 normal. And if you remember that in the history of height GWAS, the threshold where they started getting hits was when they had about 20,000 people. So nobody knows whether IQ is going to be exactly like height in terms of the distribution of effect sizes, but we have a shot. We have a shot of finding something. Okay. The error exists in this page, so this is some graphic that shows a bunch of probability of different number of hits. Okay, so now as I said, we're right in the middle of doing this genotyping, so we hope to have some results by this summer, but I don't have anything to tell you right now. Okay, let me talk about something very specific about the genetic architecture of both height and intelligence, because in the samples I'm going to talk about, we have their heights and we have their intelligence scores. And these are preliminary results. We've not published these results because we're not satisfied that the effect is really statistically significant. So everything I'm telling you could turn out to be wrong. But thinking through it, I think there are aspects of this architecture that I think are going to turn out to be correct, even though I would have to say the evidence is not conclusive yet. Okay, and this is a collaboration with some competitors of the Michigan State University Twins Project. This is ALSPAC, which is a big study centered in the city of Avon, I think, in the UK. This is another one that Plomin is on. These are collaborators from NIH, and these are collaborators from so, if there are many alleles of each of small effect, then discovering the individual loci is hard, because each individual locus only has a tiny effect. Like having maybe, the biggest single effect size known for height is such that if you had the plus allele, maybe it makes you like half a centimeter taller or something like that. So you can imagine that discovering these many different alleles of small effect is quite challenging. But if you think about that theoretically, you might consider the limit where there are a huge number of individual loci that influence the phenotype, and they all have tiny effect sizes. Okay, and surely that is very challenging for GWAS, but maybe there's some other kind of analysis you can do of the data which actually is enhanced by taking that limit, by having many, many alleles affect the trait. So imagine that the effect sizes are really, really tiny, and they're 10,000, say, causal alleles. What that means then is that somebody who is 5'5" must differ at a large number of places in their genome from someone who's 7 feet tall. Because in order to make that, to cause that difference in the phenotype, if the effect sizes individually are small, there must be, in aggregate, quite a lot of genetic differences between those two people that are specific to height. And the question is, can you, by analyzing data in a slightly novel way, can you detect that? And from that, you'll learn something about the genetic architecture. And so these are our rough conclusions, and I'll get into why we did this. So it looks to us, by doing this kind of analysis, it looks to us like the associated alleles, the alleles associated both with height and with intelligence, are at small MAF. So they tend to be rare. They tend to be at like 0.1 or 0.2 frequency in the population. So most people have one variant, and only 10 or 20% of the population are even much less having the other variant. There are more minus than plus minor alleles. So if you take the minor allele, the rare one, it's more likely to cause a negative effect. So it makes you, I don't want to be normative here, but it makes you shorter, or it makes you less intelligent, and plus makes you taller or more intelligent. And from all this, you can get a rough estimate that there are about 10,000 causal alleles, both for height and for IQ. And this could be way off. I mean, this could be out by a factor 2 or a factor 5, but it's probably not out by a factor 2. Okay, so what's the data? Alzpac is 4,000 people. We have their age 15 IQs. We have 2,000 individuals, age 8 IQs. We have their SNP genotypes. TEDS is 2,400. We have their age 12 IQs. And for ARIC, and this is like a heart health study. We have a couple more for which we just have heights. And all of these people have been genotyped using a SNP chip. We have about 500,000 SNPs for each person. Interestingly, if you just take 2 people at random out of these populations, by the way, these are all very homogeneous populations of Northwest Europeans. They tend to differ at about 261,000 loci, plus or minus about 1,500. So you can see that this is a very narrow distribution. So any 2 people from this population differ from anywhere between 260 and maybe 263,000 SNPs. But there isn't a lot of variation. Like you just won't find 2 people who differ in only 240,000 SNPs or 300,000 SNPs. This is a Gaussian distribution, and the width is just quite narrow. So what do we try to do here? The easiest way to think about it conceptually is we take some outlier groups. We take the very tallest people in the population of 4,000. We take the very shortest people. Okay? And then we calculate the average genetic distance, the average number of SNP differences between those 2 groups of people. And then we choose other groups where the heights are slightly different. And we find that as we change the relative difference in height, there is a relative difference in genetic distance. So you can ask questions like, well, how many SNPs do I have to flip in order to change your IQ or change your height by 1 standard deviation? 1 standard deviation height is like 3 inches. So how many places do I have to reach into your genome and make changes in order to change your height by 3 inches? And you can get an estimate for that through this kind of calculation. And the number is about 40. It could be 20, it could be 80, but order of magnitude, it's like 40. Okay. So that, to me, if you'd asked me, the kid reading science fiction, hey Steve, when you're 46 years old, are humans going to know like the genetic architecture well enough to answer the question, what does it take to increase your IQ by 15 points, or what does it take to make you 1 inch taller? I would have said, no way. I'm studying physics. But I'm telling you that for sure we're going to know the answer to this question in the next 5 years. No question about it. Okay, so here's a geometric picture. These are exceptional people. Maybe they're very tall. These are maybe some normal people. And we're trying to measure a systematic difference in the genetic genotypes of these people. Now, the typical distance between individuals, this distance or this distance or this distance, is quite large. And we're trying to extract out extract numbers which are quite small, like of order 100 SNPs or 50 SNPs or 40 SNPs. And so you might say, well, how can you possibly extract this from a background of this? But of course, you're averaging over 1 million pairs, so you can beat down this 1,000 by 1 over square root of 10 to the 6, and that's why you can pull out numbers like that. Okay, so here's another picture of what it looks like. So here's a group of tall people, here's a group of short people. As we vary the height, average height of this group, so we select different people, and we vary the height of this group, so we select people. This genetic distance varies systematically. And so we're extracting a genetic distance which is associated with, we're not changing any other things about these people, just their heights. And we think that that is actually then the number of SNP flips we need to actually make these people that much taller. Okay. Another thing which is more subtle is that if you ask for a group of tall people or a group of below average height people, what is the typical genetic distance between them? What we find is that the shorter people are further apart genetically, and the taller people are a little bit closer together genetically. And that actually tells us something very subtle about the genetic architecture. So let me explain that. So how do you define genetic distance? Well, the simplest way to define genetic distance is you give me a string of SNPs. And the person at each locus either has the plus version or the minus version. Okay? So here's a person with all pluses, and they have this one minus. Here's a person with all pluses, and they have a minus, but it's at a different location. So there's a concept of distance, which is used in information theory. So it's a distance between two strings. And it's basically just you sum up the number of places where those two strings differ. So the two strings differ, they differ here, and they differ here. Otherwise, they don't differ. So the Hamming distance between these two strings is 2. I've written another one down here where I've now increased the number of minus alleles. And if you look at where I put them, the Hamming distance between these two strings is 6. Okay, so there's a measure of distance called the Hamming distance, pretty standard in some mathematical applications. And that's one of the measures we use when we say that the two groups got further apart or one group is spread out more in genetic space. Now, in formulating these strings of genetic code, I've made the assumption that most people have the plus allele at each spot, and the minus alleles are kind of rare. If I were to reverse that assumption, what I would find is that, so here increasing the number of minus alleles increases the Hamming distance. If I reverse that assumption, I would get the opposite. So the fact that the people who are shorter, who have more minus alleles, are further apart, tells you it's the plus alleles that are common, it's the minus alleles that are in the population. I don't need to do that. So here's another picture. Same thing with IQ. We find the high IQ people tend to be closer together, the low IQ further apart. And that's the way to think about it is these guys have fewer of the deleterious variants. These guys have more of the deleterious variants. But the deleterious variants are rare when you spread them out over the whole genome. And so they tend to make this person more different from this person than happens in that. Okay, so that's what this says. So number of rare alleles that have minus effect is greater than the number of rare alleles that have plus effect. And in fact, it's plausible that this inequality is strong, so that the number of these is much, much greater than this. And I'll explain why that we might think that for theoretical reason. If you accept all that, then you get a very simple toy model. In physics, we often hear about a spherical cow model. So this is a kind of toy model for what's going on in these quantitative variants. So you have some number of causal variants. All the minor alleles have minus effect. So n plus is 0, n minus is n, capital N. Typical frequency, maybe, we'll just take it arbitrarily to be 0.1 for the minor allele. And then from this, you can extract the distribution, a binomial distribution, and the standard deviation is about 0.1 times n all to the 1/2. So if you back this out, what you find is that this implies that you need n, the total number of causal loci, to be about 10,000. And for that, you would get 1 SE change in intelligence per 30 or so extra experience. So in this toy model, all the parameters get fixed. If you want to make these assumptions, then you can just fix all the parameters. And basically what it means is you have 1 million SNPs, 10,000 of them are affecting IQ. Each person at each of those loci has about a 10% chance of having a minus. And that produces 30 extra minuses per change in 1 SE change. Okay. why is this, now after we were beating this data and beating ourselves up for a long time, we actually thought about it and we realized, well, this kind of has to be true just from what we know about evolution. So there's a kind of theoretical or evolutionary argument for why the toy model that I just gave you should be roughly accurate. So